Uh, Thank you for that introduction. So I'm going to shift gears a little bit and talk about some basic science and mouse immunology, specifically a molecule called 4-1BB and its role in the generation of tissue-resident memory CD8 T cells. So as I'm sure most of us are aware, influenza is an acute lower respiratory infection, and current vaccination strategies focus on inducing humoral or antibody-mediated immunity against influenza co-proteins, which are constantly changing targets. And as a result, a different vaccine formulation is needed each year in order to be effective against the new emerging strains in circulation. A potential strategy is to induce CD8 T cells for this elusive universal vaccine because CD8 T cells can target more conserved internal viral proteins and thereby maybe providing some heterotypic immunity against different strains of the flu. The problem is memory CD8 T cells appear to be fairly transient. Work in human studies Work in human studies following cohorts after the H1N1 2009 pandemic have shown that in human PBNCs, memory CD8 T cells seem to disappear after about a year. And also in mouse models, the lung seems to not be very conducive of an environment to memory CD8 T cells, as that population seems to drop after a couple of months and disappear after about half a year. So that first line of defense in that mucosal surface is gone. Furthermore, aging is also associated with the immune senescence response in the CD8 T cells, which is also problematic because over 90% of all influenza-related deaths occur in people over the age of 65. So the question we wanted to ask is, how do we achieve robust, long-lived T-cell immunity against the flu? In a way that maybe we can vaccinate people earlier on in life so that they have immunity that can continue to protect on into older life. And in our lab, we were interested in investigating this through T-cell co-stimulation, specifically a molecule called 4-1BB, which is a traf binding member of the TNF receptor superfamily. And by way of MAP kinase pathways, there we go. By ERK and MAP kinase pathways and multiple signaling pathways, it's implicated in T-cell survival as well as cytokine production. And its expression is induced upon TCR signaling and can be sustained if the antigen is present. Our interest in this molecule comes from the fact that previous work in our lab has shown that mice deficient for 4-1BB ligand, which is the cognate ligand for this pathway, have impaired recall responses to flu, possibly due to a loss of the effector memory population after primary infection. So with that in mind, we ask, can we potentially exogenously stimulate 4-1BB as a way of stimulating the CD8 T cell response against influenza? So we used a replication defective adenovirus 5 vector that contains the NP gene from influenza as the antigen, as well as 4-1BB ligand. And this was delivered in a very localized way by intranasal infection. And the significance of that I will address a little bit later on. And this also allows for a very sustained delivery of the antigen with 4-1BB ligand, as the adenovirus is known to persist in antigen presentation for more than 40 days. And a previous postdoc in our lab gave this intranasally to mice and found that this ADAMP4-1BB ligand was able to enhance CD8 T cell memory responses and protect mice for about 3 months. Which is nice and all, but 3 months really isn't a very long time. And given the body of literature showing that 4-1BB is a more potent co-stimulator of the secondary CD8 T cell response, we decided to give it in the boost phase of a prime boost immunization regimen. So this is our model. We infected mice with a mild flu infection, waited for memory T cell responses to occur, and once that was established, we boosted these mice intranasally with either add with the MP antigen alone or add with MP and the 4-1BB ligand. We also had mice that were left unboosted or boosted with control adenovirus as controls. And the rationale here is that yes, we are boosting for that secondary immune response, but flu is very prevalent. Most of us have probably come into contact with it already and have some sort of immunity to it, that this would mimic the vaccination of influenza immune adults, which most of us would probably be. So for a first set of experiments, we waited 9 to 11 months, and then once these mice were older, we challenged them with a lethal flu, just to test that protective efficacy. During that 9 to 11 months, we decided to track this memory response and see how 4-1BB is affecting these cells. And we find that very early on, there's a very dramatic enhancement of memory CD8 T cell responses. And this is measured by tetramer, that's for the NP epitope in flu for CD8 T cells. And this is occurring at a dose where ADAMP alone really doesn't seem to have much of a response compared to the two controls. This is looking in the blood of these mice. What's really interesting to us is that it's not only a robust response, but it's also a very long-lived response. Looking out to over 300 days post-boost, we still see a fairly robust population of these cells. And that's something you can see here as well in our representative gating. And that's recapitulated as another way of measuring the systemic memory population in the spleen, where we've enumerated the number of memory CD8 T cells that are NP specific, and we still see a large population with the 4-1BB ligand boost, whereas ADMP really doesn't have anything at like 7 months post-boost, fairly long after the administration. Well, that's nice and all, but what does it really matter? Well, about a year in, once these mice are fairly aged, we gave them a lethal challenge of PR8. And PR8 is a good virus to use because it contains the same NP epitope as X31 or initial priming virus, but because it's H1N1 compared to H3N2, this allows us to look at the CD8 T cell responses without too much interference from neutralizing memory antibody responses. And when we did that, we found that 4-1BB ligand boosted animals were protected from influenza-mediated weight loss, and in fact survived the lethal challenge with about 80% survival, whereas without 4-1BB ligand, the ADMP group is only really able to protect 40% of these mice. What's also interesting to us is that 4-1BB stimulation is associated with a lot of cancer therapies, and where essentially they would give you a systemic administration of anti-4-1BB agonistic antibody, and that's resulted in a lot of liver toxicity and immune pathology. And that's not something we see in our model, because we think that this localized administration is keeping that stimulation contained in that respiratory tract where influenza impacts. And therefore, we get that survival advantage without the pathological consequences typically associated with systemic 4-1BB stimulation. So with that in mind, we next asked, well, how are these memory cells doing this? How are they any different? And to not take up too much time, I've summarized a couple of things that we saw with the systemic circulating memory population. And the take-home message is that they were very effector-like. And some of those characteristics are that they have low CD62L expression, they're low in EOMES, TCF1, and high in TBET, which are characteristic of effector memory populations. And they also have greater effector function as measured by interferon gamma, TNF-alpha, as well as CD107A, which is a degranulation marker. Interesting to us also is that they express high levels of CD127, which is the IL-7 receptor. And IL-7 is known to be important for the survival and maintenance of memory CD8 T cells. So this may contribute to that longevity of the response that we see. And all of this does have implications for protection because effector memory cells are more in the periphery in their circulation and can respond more immediately and more robustly to an encounter with antigen. But that's nice. But what we really want to know is what's happening in the lung, because that is the mucosal surface of infection and it's the first line of defense. So what we did is an in vivo staining technique where while the mice are still alive, they get an in vivo injection of, in this case, anti-CD8 antibody. So we stain them with EE, for example. And all of the cells that are in the vasculature get stained. And then afterwards, a couple of minutes later, we sack these mice, we process the tissue, we take the lungs out, and we stain it again with another CD8 antibody in a different color. And what we get is a differentiation between cells that are in the vasculature, that are traversing through the lung tissue, versus those that are actually within the lung tissue. And what we see, and this is looking at 7 months post-boost, a fairly long time point, that you pretty much don't see any CD8 T cells in the ADMP boosted groups at all inside the lung tissue, let alone any memory cells. But if we look at the 4-1BB ligand boosted groups, we see a very big population of memory CD8 T cells that still persist in the lung. And it's a fairly stable population as it seems to remain about the same number between day 60 and day 200. So it seems to be very well maintained and long-lived. Through a series of mixed bone marrow experiments, we also determined that this is in fact 4-1BB ligand acting directly on 4-1BB that's expressed by the CD8 T cells themselves, suggesting a CD8 intrinsic mechanism in this local and systemic expansion. What's also really important is that the route of administration seems to determine this lung resident memory response. So we took mice, infected them intranasally with influenza, as we read before, X31, and then boosted either as before intranasally or intraperitoneally. And what we find is that if you look at that ADMP4-1BB ligand boosted group, intraperitoneal systemic administration of 4-1BB ligand does not induce CD8 T cell lung accumulation within the lung tissue. And that's fairly consistent with what's in the literature, where the route of administration, that local immunization, is in fact crucial to the establishment of local resident memory responses within the tissue. And then with experiments that we did with a drug called FTY720, which blocks lymphocyte exit from lymphoid tissues and keeps them sequestered, we found that 4-1BB ligand is in fact inducing a recruitment of memory CD8 T cells into the lung tissue from the circulating population. So everything I've shown you so far is very much an exaggerated exogenous stimulation of 4-1BB in this model. And what we wanted to know is, what is 4-1BB endogenously doing in the flu response? So we generated mixed bone marrow chimers where essentially we took bone marrow from 4-1BB competent mice, bone marrow from 4-1BB deficient mice, and we put them into a 1-to-1 ratio into wild-type irradiated hosts. And the idea here is that these irradiated hosts are getting a complete reconstitution of their system, where we will get CD8 T cells that both have 4-1BB and CD8 T cells that don't have 4-1BB. But they're in the exact same environment where the 4-1BB competent donors has provided all the other cell types that need to be 4-1BB competent. So in this case, we're trying to assess, is there a direct and intrinsic role of 4-1BB on the CD8 T cell compartment? And what we did was very simple. We let these mice rest and reconstitute, and then just gave them a mild flu infection and looked 30 days on to see what's happening with the memory responses in the tissues of question. And when we look at what we plotted, which is a ratio of 4-1BB sufficient to 4-1BB deficient cells, we see that if we look at the empty bars, which are just regular CD8 T cells, not flu specific, it's still very much a 1-to-1 ratio over all of the tissues, suggesting that there's no survival advantage of either of these cell types. But if we look at the lung tissue of the flu-specific memory CD8 T cell responses, we see that it's pretty much completely consistent of 4-1BB sufficient memory CD8 T cells, suggesting that if CD8 T cells don't have 4-1BB, they don't contribute to the memory pool within the lung at all, whereas it's still very much 1-to-1 in the draining lymph nodes and the spleen. What I won't show you because of time is that this dichotomy is something we see at day 30. We don't see it at day 9, and we don't see it at day 18, suggesting that 4-1BB is having that critical CD8 intrinsic role in the maintenance of lung TRMs, tissue resident memory cells, as opposed to the formation of these cells. So a quick summary of everything I just told you. We gave a exogenous stimulation of 4-1BB in an adenovirus model, importantly through the intranasal local route. And what we got was a direct interaction with 4-1BB that's expressed on the CD8 T cells to mediate a secondary CD8 T cell response that's characterized by a long-lived effector memory systemic population, as well as a long-lived tissue resident population, both of which are coming together to mediate a protective response against the flu. And through my last slide, I showed that the 4-1BB ligand pathway is in fact acting directly on CD8 T cells in the lung to critically regulate CD8 tissue resident memory maintenance during influenza infection. And why is that important? While realistically no one's going to generate a vaccine that has 4-1BB ligand in it, it's important to understand co-stimulation in the antiviral T cell response, because we need to know what signals need to be activated in order to generate a protective response against certain viruses. And that is very much important for informing adjuvant design in the future of vaccine development. And with that, I would like to conclude and thank my supervisor, Dr. Tanya Watts, everybody in the Watts Lab, our flow facility, as well as supervisory committees and people involved.
Thanks, Angela. That was really good. So we've got time for a couple of questions. So there's one. Oh, there's two. So the first is there a chance for the migration of these memory cells to other tissues where they can have off-target effects?
We haven't really seen that, but to be fair, we also haven't really assessed too many. We haven't really assessed too many other tissues aside from the ones that are involved in the respiratory influenza pathways. I would think maybe not, because the local inflammation in the lung is what is recruiting cells into the lung. So we haven't really looked, but I don't really think that that would be a problem.
Okay. And second question is, why would 4-1BB not be in a vaccine formulation?
I think to put in a ligand that directly stimulates a molecule may not necessarily be the best idea, because we may not see it in our model, but I think it could potentially have off-targeted effects. I think what's more important is that there are a lot of TNF receptor superfamily members like 4-1BB, and they all kind of come together to uh, induce different effector responses. And it's important to know how they're induced and maybe put certain signals in that will upregulate these molecules and have that be a much more natural kind of an antiviral response versus actually putting in a ligand.
Okay. And I think we've got time for one more. You're very popular. Did you see any impact on central memory CD8 T cell populations?
What we actually saw was a skewing away from central memory T cells into more effector memory cells. So I understand that the paradigm is a little bit different between mice and humans in that in the mice model, there's a slow conversion of effector memory to central memory, and that the central memory is the more stable population in the mouse, whereas in humans it's a little bit different. But what we saw was more, it seemed like they were converting much slower to central memory, and that they were being maintained in that effector memory phenotype a lot longer.
Okay. One more question.
Okay.
Oh, no, sorry, I don't see it. I don't think there's any more. Somebody might be typing. Okay. I just wanted to thank you. Okay. Thank you so much. That was great.